So in a follow-up to our previous Diels-Alder lecture, we now have kind of our big reveal on what happens with the Diels-Alder reaction. So the first two slides here are just to kind of give you a reminder. When we're looking at the Diels-Alder reaction, we are starting with a diene, so named because it has two pi bonds, okay, two different pi bonds. And we have the dienephile because it likes to react with dienes, so it likes to interact with the diene structure. The net result of any Diels-Alder reaction, or most Diels-Alder reactions, is you get this cyclohexane-like structure, so a 6-carbon ring and one double bond remaining within it. If you change the characteristics of either the diene or the dienephile, you can change your base structural result. So the classic example would be if our dienephile was indeed a triple bond. And I'm attempting to make that look nice, so let's just erase it. 1, 2, 3. We still have a dienephile, so that can react, that reacts, and pi bond shifts, so we still end up with the pi bond, except we'll have a leftover pi bond on the other side. Okay, so just be aware that Diels-Alder reactions are looking at pi bonds, not necessarily alkenes. It just seems the alkene is most obvious. So that's our basic structure behind this. The next part is identifying the things that changed. So we could look at things that were broken and formed, and what we ended up talking about was that really we can simplify this down to saying the only thing formed were these two sigma bonds, which we labeled as sigma 1 and sigma 2. Within that context, we then said, okay, if those were really the only things that changed, then how did those changes occur? Did they happen sequentially, or did they happen simultaneously? And that was kind of the point of our lab. So in that previous lecture, what we went through and did is looked at the guess mechanisms. So we came up with two possible mechanisms that we labeled DA1 and DA2 for Diels-Alder 1 and Diels-Alder 2. DA1, just like our SN1 reaction, is defined as being stepwise, and the primary result of that is that we end up with two different products. Okay, so that's our big issue with it, or a big result of the DA1 mechanism. If we move to the DA2 mechanism, since it's all happening at the same time, we only end up with one product. Okay. I also ended that lecture with this kind of image of the model kit in the lower right, and said that you need to play around with that model kit to get an understanding of how these reactions are occurring. That will become increasingly more relevant as we go through and look at the end results of the Diels-Alder reaction. So I'm going to remind you again, if you have not yet done or played with the model kit to see how these mechanisms result in these products, you need to stop the video now and play around with it and get comfortable with what's happening in the process of that. Okay? Those of you that have already done it, let's go ahead and move on. So what we do is we go to the lab, and we have to remember again our initial theory on this, and what we're ultimately testing is, do we get rotation about the diene bond? If there's rotation in the course of the mechanism, that means I have a sigma bond that is free to rotate, and I end up with two products. So rotation leads to DA1, and no rotation leads to DA2. And so what we might predict out of this is that if there is rotation, we would expect two products. If there's no rotation, we would expect one product. So we go into the experiment and we test. So one of the things we'll do is try and keep things as simple as possible. So you'll notice, since all of my focus is on the dienephile, I'm keeping the diene constant and very, very simple. We don't want the diene messing with our reaction. So I then mix this with my dienephile, and I've got two symmetric groups on it. And again, we want symmetric. We don't want to mess with anything. We're trying to keep our variables as simple as possible. So in this case, I'm using the cis alkene. And when I go through and run the reaction, what we end up with is one product. Okay, so this OR is here to show you different ways to view this molecule. The way we've been talking about it and looking at our mechanism is that we're showing the three-dimensional characteristics by showing this kind of upside-down boat form. Or we could look at it in an overhead view. So this is our overhead. And what you would notice is that if our eye was looking from above, those carbonyl groups are now aimed away from you, hence the dashes down. So we ended up with this one product. So this starts to suggest which mechanism is driving our reaction. Hopefully you come up with the Da2, that it's all happening in one step. Well, this is only one piece of evidence, so ideally we want to make a change and see what happens. Maybe there is something about the cis nature of that starting alkene that is going to mess with our reaction dynamics, and it's going to now force those to be on the same side. Okay? So we want to make sure that we don't have to worry about that, so we'll run a second experiment, and that second experiment will now switch to the trans Diene file. Okay? And then we'd see what happens. So we go to lab, we test it, we work it all up, and we end up getting one product. Okay? Again, suggesting Da2. What kind of interesting things start to fall out of this? Well, if we look at experiment 1, we started with a cis alkene. What is the relationship of the groups in our final structure in experiment 1? Well, hopefully you see that they are cis. They are on the same side of that ring. When we moved to experiment 2, we started with the trans alkene. What happened when we got into our product? Well, you should again hopefully notice that what we end up with is the trans product. So the relative stereochemistry, if you will, of our starting material is then forced into our product, and we see no bond rotation. This means that the Da1 mechanism is entirely invalid, and all we're looking at is the Da2. This should make you kind of happy, because that means you only have to worry about one mechanism. Okay? Just in case you missed it again, with our final product in experiment 2, same deal as with experiment 1. This is the overhead view on the right, and then the middle one is showing our stereochemistry. Well, it turns out that these experiments were run multiple times, multiple different people, and they all ended up coming to the exact same conclusion. Regardless of how you change the rest of your structures, you still end up not seeing this bond rotation. Da1 does not exist. Everything must happen at the same time. So we could say same time, we could say Da2. The 2 doesn't really make any sense anymore, so we just end up calling it Diels-Alder, because there's only one mechanism. But it still wasn't scientific enough, so the term that we end up coming up with is concerted. All of these things happen at once. So if we go back to our reaction, if we want to draw in mechanism arrows for experiment 1, what we are saying is that the diene shares its electrons with the Diene file, which then causes those electrons to come out and share with our diene, which then causes the next pi bond to shift. So we get 3 arrows all happening at the same time. Take a minute, let's pause the video, and I want you to go through and do it on experiment 2. Add your curved arrows. And hopefully you're back. So we've got our diene sharing our electrons. In this drawing, it may be easier to see that we can take those electrons and we share it with the alkene. A little bit more straightforward. However, where does that next pi bond move to? So this is one of the confusing things. You may say, well, it looks really close to this, so maybe it goes down and does something weird like that. Unfortunately, that weird should be a big tip-off. That's not the case. What that alkene then does is shift over to our diene. Okay, so the orientation may look a little bit weird for our drawings. So that's fine. We want this orientation. We'll talk about that a little bit later. Okay? So that's ultimately the result of all of that mechanistic mumbo-jumbo that we spent 20 minutes on last time, or 40 minutes on last time. is getting to the bare bone detail of this. And hopefully when we move to the lab for our class, we'll see the exact same result. So, can we make this more complicated? Well, in both those cases that we just looked at, the only thing we've touched was the Diene file. What happens if we mess with the Diene? Now what we've done is change the structure around a little bit. Notice we're still maintaining symmetry on the Diene file. We want to maintain that symmetry because we're trying to help simplify our problem. Okay? And as we continue to work forward, we'll keep removing that symmetry and make it more and more asymmetric. But at this point, it's symmetric. So what we can go through and do is add our curved arrows. So go ahead and again, pause the video for a minute and add curved arrows and predict your products. And we're back. So we'll take our electrons, shift across, shift across, shift across, and if we draw out our result, we've got our basic skeleton, the double bond on the left based on our drawing. We still have that methyl group, and then we have the carbonyl and our other carbonyl. Okay. And I'm actually going to draw the other product here. So we can go through and draw out that one. Same idea. As soon as you think you got a Diels-Alder, what you want to do is immediately start drawing in that cyclohexene. Okay. Hopefully you've got these basic skeletons down, and that's a very good first step, and that might help eliminate answers on a multiple choice test. But we now have another issue to worry about. What you should notice is that in the course of this reaction, I now have 3 stereocenters in our red product, and I have 4 stereocenters in the purple product. So that means we have to specify them. Well, we may have already kind of identified some of this. So you may be able to address the carbonyl functional groups. Okay, so let's go through and do that. Well, in both cases, these were cis alkenes as starting, so we need to make sure that the carbonyls, be they wedged or dashed, because remember your Diels-Alder reaction can happen either above or below, whether wedged or dashed, they must be the same. So what I'm going to go through and do is on the red one I'll draw dashed, and on the purple one I'll draw wedged. As long as it is consistent within the product you are drawing, you're fine. Okay? So that helps us out. We've now nailed down some chemistry and some arrangement of those groups. We may have just eliminated a couple more answers. Okay? But almost undoubtedly, in the case of our red one of those answers will show that red one as a wedge, or that last stereocenter as a wedge, and one will show it as a dash. Okay? How do we decide which one it becomes? Well, it's ultimately going to come. Remember I said you had to play around with those model kits? It's going to come to how that group started relative to this attacking group coming in. Remember, what we're looking at is kind of a side view. Let's see if I can draw the side view real quickly. And if we look at that methyl group, it's out here, and we have a hydrogen on the inside, and our diene is coming up, or our Diene file is coming up this direction. So as it comes up, okay, whoops, should fix my arrow, sorry. As it comes up to form sigma 1 and sigma 2, this hydrogen is going to have to move. Because if we just left it as it was, it would be stuck in the middle of the ring structure. And that makes no sense. Okay? Well, if the pi bond is coming up towards that hydrogen, where does that hydrogen necessarily have to move? Well, it's got 2 choices, and this is going to be hard drawing in 2 dimensions. It can either rotate up, or it can rotate down. If it rotates up, it's rotating up through empty space. If it rotates down, it's now rotating through our diene, or our Diene file. That's a bit problematic. So that hydrogen cannot rotate down. It must rotate up. Well, if it rotates up and this is our overhead view looking down on this, we would show that hydrogen being wedged. Well, if the hydrogen is wedged, what does that mean? That last methyl group must be in our product. It must be dashed. Incredibly hard to see, even harder to draw, much easier to see if you use the model kits. So I highly recommend you pull out the model kits and you view this, because if you have the model kit in front of you, you will be able to see this orientation. It may take a little bit of practice getting used to. Talk to me if you want help with that. What happens in the next one over? Okay. Well, if we run this same kind of idea, let's see if I can clean up the drawing that I've got. If we run this same idea, so now we're looking solely at our other system. What we've got over here is our hydrogen. Here's our alkene. The alkene comes up, and now instead of the hydrogen being in the way, the methyl group is in the way. Whoops. Shoot. Shouldn't have redrawn everything. Okay, so let me redraw over here. Let's see if I can do this right. We have to angle this down because I conveniently drew the product where it was wedged up. Okay. As these come in, as we form that sigma bond, where does that methyl group rotate? Okay. Being on the inside again, it's going to have to rotate down in this case because we drew the opposite viewpoint, the opposite sigma bond formation, away from the forming sigma bond, which means it must be dashed. Okay. How is that dash different from our red product? Okay. It is different because if we look at our red product, where are our carbonyls? They're dashed. So in our red product, they're all on the same side. In our purple product, you'll notice that they're on opposite sides. What happens with the other methyl group? Okay, and again, we'll try and clean this up. Okay. Well, the group on the inside had to rotate down into open space, which means the hydrogen is now aimed, just like our methyl group was, away. Well, if it's away, what happens to the methyl group? The methyl group ends up being aimed towards, which means it must be drawn wedged. Okay, clear as mud. Beautiful, isn't it? Okay, incredibly difficult to see. Okay, one of the things that'll make this even more complicated is, remember, because you can form sigma up and sigma down, we have two approaches for our diene-dienophile interaction. We end up with enantiomers. So if we pull this out, there's the sigma down. They're all on the same side. Sigma up, you'll notice that, or sorry, the sigma down over there, you'll notice that our methyl groups are aimed on opposite sides of the ring. One of them is on the same side as our carbonyls, the other one is not. Okay? And remember, because of the enantiomers, actually, before we do that, if we look at the sigma down pattern all the way through, we've got our starting materials now labeled with these individual positions. Okay? So that if you look at the proper orientation of those, you can now predict where those groups go in the final product. So you can either be comfortable with the three-dimensional shapes and how these things are interacting, or you memorize this little sigma down thing that I've got at the bottom. The problem with the memorization is you also have the sigma up product, and you'll notice things shift slightly around on you. So you got to be a little bit careful with that. Okay, the products that I drew for the sigma up, I tried to draw in such a way that you could see that they were enantiomers to make that visualization easier to see. Okay. Practice with the model kit will help you nail down these proper orientations and where these individual groups go. It takes time. I will fully admit that when I take a Diels-Alder test, I try to skip these questions, draw out the skeleton, and if I got time, I'll go back and spend some time to nail down the exact stereochemistry. If not, I'll just take a guess at it. Okay? The easiest ones to nail down, remember, would be the orientation of your Diene file. Okay? If it starts cis, they'd better be cis in your product. If it starts trans, they'd better be trans in the product. After that, it just takes time and practice. So, this whole sigma up versus sigma down was a bit irritating, I'm sure, for you. What if there is no sigma up and sigma down? So you'll notice that the primary change that we've gone through and made in this case is that we took the diene and I added this carbon as a bridge between those two atoms. That now locks the structure in place, and it has this extra little feature that if we grab this carbon and pull it up towards you, you end up with the viewpoint we have here. And what's even more interesting about that is that if I now try to form the sigma up and the sigma down, this carbon blocks the sigma up orientation. So I don't get a sigma up product. Just kind of nice. I now lose that potential for an enantiomer kind of deal coming out of it that we saw in our previous ones, which is kind of a neat system. Well, it turns out that there's a problem with this thought. Okay? So take a minute, pause the video, and say, is there another way that we could run this reaction where both of these products can coexist? Okay? And this becomes a little bit tricky. Look at the structures that I've got drawn, okay, of both the diene and the Diene file, and is there another way that you can orient those to generate both of those other products? So the change that we can make would be the orientation of the Diene file. You'll notice that in all cases, or both these cases, I've shown that oxygen, if we call that an arrow, pointing towards the left. Did I have to show it pointing to the left? I could have changed that orientation such that what I had was something along these lines. I can change it so it's pointing the other way, and now I can still do the sigma down, which would necessarily result in that product. You should be able to prove that to yourself. So, ouch. We hoped that we could get rid of sigma up and sigma down, and instead what we do is introduce a whole other problem. And this problem was well studied before, and it gets referenced by these different names. We end up with an endo orientation and an exo orientation. Whatever you can do to come up with something to remember that is kudos to you. Endo, for me, I tend to think kind of in or under, so you'll notice that these are kind of now stacked on top of each other, whereas the exo is on the outside. If we tried to draw that same kind of square, you'll notice that we have a piece outside that square. Okay, so we have an exo orientation or an exo product and an endo product. So, since we now have two possible products, we want to go through and evaluate and see what the differences are. So, number one, are they enantiomers? Interestingly enough, they are not enantiomers. And that's because of all this extra ring constraints that we put on this. What we've got are diastereomers. And if they're diastereomers, they have the potential for being different energies. So we want to evaluate now the different dynamics of those energies. So, if we go through and look at some of these results, the endo is a sterically hindered product. Well, what do we mean by sterically hindered? Well, I've got this 2-carbon bridge near all of this electron bulk. So we got electron bulk butting up against electron bulk. That's bad. So the product energy is going to be higher in energy because of this steric hindrance. If we look to the exo product, you'll notice that now when we look at our steric bulk, we're looking at a large group near a small 1-carbon bridge. So while there is some steric bulk there, it's relatively smaller than in our endo product, which means we should get a lower energy product for our exo. Okay? That would suggest the exo product wins out. Well, of course it can't be that simple. We've got other stuff going on as well. So if we look at our products, now we also have to shift to look at our transition states. So in the transition state, what we're referring is what happens right the instant before the bonds are formed. If we take a look at the endo, we have p orbitals on this guy and this guy that can stack with those p orbitals. Okay, and this is kind of a debatable thing coming up here, but this p orbital stacking, or pi orbital stacking, is a known feature. It's one of the reasons why DNA is incredibly stable as well. This stacking lowers the energy of the transition state for the endo product. When we look to the exo, you'll notice that our p orbitals are nowhere near each other, so they don't get this favorable lowering of the transition state. So we now have a competition. We have a thermodynamic issue looking at the energy of our products, and we have a kinetic issue looking at the transition state. So which one ultimately wins out? Okay. Well, that exact decision would come down to experiments and different ways that would control that, and we have talked about that last semester. In this case, what we end up finding out is that for the Diels-Alder reaction, almost exclusively we get the kinetic product. It's almost always endo. Very little exo product actually shows up. Okay? So let's shift our viewpoint to an energy diagram now and evaluate this, walk through it, okay, so that we can all be on the same page here. Our reactants are ultimately at the same energy because it's the same reactants for both. Okay, so, and since we don't necessarily know where anything else is, we're going to just kind of float that in there. So there's the energy for our reactants. Purple, because our reactants for both the red reaction and the blue reaction now mix, or at the same energy. If we now look to our product, okay, well, our lowest energy product is going to be the exo product, okay, because it has less steric bulk, which means I should draw it relatively low. I would also expect it to be lower in energy than our reactants because we've lost a couple pi bonds. And if we look at our red product, it should be higher in energy than our blue, but we still lost pi bonds, so it should be somewhere in there. Okay, we don't need exact values, but it gets us an approximation. The next part is moving into our transition state. Okay, well, for our endo product, we're saying that the transition state is relatively low. And since I'm kind of out of space, okay, it should be higher than our reactant energy. Okay, and our transition state for our blue is relatively high. So if we now go through and attempt to connect, we get something like this for our reaction. Okay, so there's an activation barrier for both of these, even though it looks a little bit weird. Okay, and so what happens is that we would expect at relatively low temperatures, which means low energies, that the only product we would expect to get would be our endo product. Okay, because we don't have enough energy to clear the activation barrier for the exo product. So even though that exo product is lower in energy, if I can't get over the activation barrier, I can't make that product. Okay? So we'll keep kind of thinking about this when you think about energy diagrams and what product is favored. This is really what you should be trying to do, is draw out some version of an energy diagram and come up with an approach. Okay? Next part of this. Now that we know the basics of the reaction, let's get out of lab faster. What can we do to speed the reaction? Okay, well, in previous circumstances, what we did to speed a reaction was we made the nucleophile more nucleophilic and the electrophile more electrophilic, or those were our two options. But if we're now looking at a concerted mechanism, we've got a problem. If it's all happening at once, what's the nucleophile, what's the electrophile? Because we could go through and say, well, this arrow is nucleophilic because it's sharing electrons, but then what is the arrow down here showing? Well, it's also showing sharing of electrons. So we have two nucleophiles and two electrophiles. This is just weird. Okay, so what we need to do is kind of simplify our approach here. What we're really looking for is the best or most likely nucleophile in each case, okay, or electrophile. To me, it's a little bit easier for all sorts of weird, odd reasons, that the nucleophile is a little bit easier to find. Well, the definition of a nucleophile is an electron donor. Which of these two species has more electrons to donate? Well, the Diene has more electrons. It is very likely to be the nucleophile. Okay, so if I want to speed it up, I want to make it more negative. So this guy needs to become more negative, and this one needs to become more positive. So what I want to do is change those structures. Maybe put something out here that has a bunch of electrons that could donate in. That structure now becomes more negative. If I want to make my dienephile more positive, I want to put something out here that draws that electron density out to make it more positive, more likely to react. Okay? So what we'll go through and do is look at adding anything, in the case of the Diene, that has more electrons than a hydrogen atom. And we pick hydrogen because hydrogen is actually already on the structure, and so it's something that we're already trying to evaluate. So it's all in comparison to the hydrogen atom. So anything with more electrons than hydrogen. Believe it or not, most things have more electrons than a hydrogen atom, so that works out great. We can pretty much attach anything to this, and the Diene and our Diels-Alder reaction will react faster. Well, what about the dienephile? Okay, well, we still have that hydrogen present on our dienephile, so what can we do to speed from its end? Well, we want to make sure we have any group with less electrons than a hydrogen atom. And that becomes a little bit of an odd definition. So let's evaluate what a hydrogen atom is. And everybody goes, well, hydrogen is, H+. No, hydrogen is not H+. That is hydrogen ion. A hydrogen atom is a hydrogen with a single electron. So H+ actually has less electrons than a hydrogen atom. How could I get carbon to have less electrons than a hydrogen atom? Well, what if it's only attached to 3 things? I now have a carbocation. That, by definition, has less electrons than a hydrogen atom. That is why it is positively charged. So any atom that carries a positive charge is an electron withdrawing group. Okay? Most cases you'll find your electron withdrawing groups are sneaky and not obvious. Okay? So we've got a couple electron withdrawing groups and donating groups drawn as examples. The nitrogen is a perfect example in that last one. The nitro is your classic electron withdrawing, because what charge is the nitrogen? It is formally positive. But what about the next two? Neither of those are formally positive. But in one case, you have extremely electron withdrawing groups, or electronegative atoms, that pull all the electron density away from that carbon, making it become electron withdrawing. And what about the very top example? Well, we can draw a resonance structure that puts a formal positive charge on our carbon. There's our result. Okay? So we can adjust the speed of the reaction by changing the groups that are on our alkene, on our Diene and our dienephile. Which gets us to the very last effect. Wait a second. I didn't think I was going to give you those answers right away. If we now add in all of the asymmetry we possibly could. You notice on the Diene, I have an amine group. Okay? And it is not mimicked on the other side. Okay, so it is asymmetric. If we look at the dienephile, same deal. I've got it asymmetric. There is nothing on the other side of that to help with that balancing out. This means that when we go through and run the reaction, there are 2 orientations to produce products. So I can have these electrons come out, come in, and form what we'll call the red product. Or we could do it in the blue orientation, and we get that bottom product. 2 possible products. Are they equally favored? Okay, we'll spend a minute here, think about it, pause the video, come up with your guess. And when we come back, What do we need to evaluate to help us figure this out? Well, there's all sorts of different things that we could look at. Probably one of the ones you thought of first would have been sterics, but before we look at sterics, we need to look at electron effects, which means our favorite four-letter word, resonance. Draw out the resonance structures for your starting materials. See how that forces an alignment within the individual reactions. And what you should discover from that resonance balancing out is that you end up with really only one product, and that is the top one. Sorry, I don't want to draw the answer, I want you to draw through it. And yes, it is the top one. Okay? So, verify that. Make sure you believe it. If you don't believe it, talk to me in class. So, in conclusion, basic skeleton for our Diels-Alder reaction, already provided there up top. Our diene is more commonly our nucleophile. Our dienephile tends to be the electrophile. Be aware of your stereochemical relationships when you go through and run the reaction. Note that in this example I am specifying where these are coming. So the result of this reaction would produce the sigma down product, as we've been referencing it earlier. We have stereochemistry popping up as being a possibility depending on the symmetry of our structure. Okay? So that can result in enantiomers, and if we've got cyclic starting materials, we tend to start to push towards diastereomers. Okay? And then your regiochemistry, how or where the sigma bonds form, or the groups around our diene and our dienephile form sigma bonds relative to each other, is defined by resonance. So draw out your resonance structures. That will help you predict the proper arrangement. Okay? And an example that I gave to another student, think of it as orienting two people in space. You can orient them head to head, foot to foot, or you could orient them head to foot and head to foot. Two possible orientations. If you now ask them to make out with each other, one of those sets or one of those orientations would be better or more favorable than the other, assuming no fetishes. Okay? So, if you got questions, talk to me in class or in office hours.